Good evening. Welcome to Rest Academy. In this session, you'll get a 2-hour sleep-friendly tour of your immune system. How does your body know what belongs and what does not? What stands guard the moment barriers are breached? And how can a single drop of blood packed with thousands of immune cells already decide friend from foe? During this video, you'll relax while understanding the whole map in one calm, continuous flow. To make this journey complete, I've prepared a free practical guide with a concise summary of tonight's lesson, waiting in the description like a map you can return to whenever needed. Let's begin. You stand above a city at dusk, its perimeter lit by a ring road, avenues converging toward quiet hubs where training, sorting, and deliberation happen. This city is your immune system, a coordinated network that preserves you by distinguishing self from non-self and harm from harmlessness, then acting with proportion. Immunity means the capacity to resist biological threats. Its core functions are recognition, response, regulation, and memory. Recognition identifies patterns of pathogens or damage. Response deploys cells and soluble molecules to contain and clear. Regulation restrains actions to limit collateral injury. Memory encodes efficient future defense. Each function is real only because of where it lives. Organs and tissues that create cells, educate them, and stage encounters. The map divides into primary and secondary lymphoid organs. Lymphoid refers to structures that support lymphocyte development or activation, lymphocytes being B and T cells that form the adaptive arm. Primary organs are bone marrow and thymus. Bone marrow is a soft tissue inside bones where hematopoiesis, the formation of blood cells, occurs. Here, common stem cells give rise to all leukocytes, including B cells, neutrophils, monocytes, and others. The thymus, seated behind your sternum, is where T cells mature and learn restraint. Secondary organs include lymph nodes, spleen, and mucosa-associated lymphoid tissue, MLT. These are the city's squares and courts, places where antigens, recognizable molecular features of microbes or abnormal cells, are displayed to lymphocytes to start specific responses. On the ring road, you find tolerance as a gatekeeper. Tolerance is the state in which your immune system does not attack your own tissues or steady partners like your microbiota. It exists in two broad layers. Central tolerance occurs during development. B cells in bone marrow and T cells in thymus undergo tests that remove or redirect clones that bind strongly to self. Peripheral tolerance operates after deployment. Regulatory cells, checkpoint receptors, and carefully controlled cytokines suppress responses that might veer toward self-reactivity. Defense is the counterpart, the ability to detect and remove threats. Healthy immunity commits to defense only after tolerance has reduced the chance of friendly fire. Consider the city's patrol units. Innate immunity is the fast, non-specific police force that recognizes conserved patterns shared by many microbes or by damaged cells. Neutrophils, macrophages, dendritic cells, natural killer NK cells, and soluble systems like complement move early. Adaptive immunity is the specialized judiciary and elite responders that recognize very specific molecular shapes through receptors generated by gene rearrangement. B cells produce antibodies, T cells coordinate or kill with precision. Innate and adaptive arms interlock. Innate signals set the tone and location. Adaptive effectors bring refinement and memory. At the city's intersections, you see antigen, a molecular fragment that can be bound by receptors. Alone, an antigen is insufficient. Effective activation usually needs context, signals indicating danger or damage, so that the same molecule in different settings leads to different outcomes. This context is supplied by pattern recognition receptors on innate cells, sensing pathogen-associated molecular patterns or damage-associated molecular patterns. These signals induce co-stimulatory molecules and cytokines, shaping whether an encounter yields activation, anergy, functional silence, or tolerance. Within bone marrow, hematopoietic stem cells differentiate along myeloid and lymphoid lines. Myeloid progenitors generate neutrophils, eosinophils, basophils, monocytes, macrophages, and dendritic cells, as well as megakaryocytes and erythrocytes. Lymphoid progenitors generate B cells, T cell precursors, which then migrate to thymus and NK cells. This developmental pipeline matters because diseases that block early steps cause broad immunodeficiency, while selective blocks yield narrow vulnerabilities. Location imprints function. Marrow niches provide growth factors. Thymic cortex and medulla expose T cells to self-peptides in controlled gradients. Lymph nodes organize B and T zones to optimize chance meetings with matching antigens. The spleen, tucked under your left ribs, filters blood, not lymph. Its white pulp harbors immune cells. Its red pulp clears aged red cells and opsonized pathogens. Asplenia, absence or loss of splenic function, raises susceptibility to certain encapsulated bacteria because the spleen excels at mounting rapid antibody responses against them. Lymph nodes filter lymph, the clear fluid draining from tissues, routing dendritic cells bearing antigens to T cell areas and naive B cells to follicles. Mount, scattered along mucosal surfaces like gut, GLT, airway, BLT in some contexts, and nasopharynx, tonsils, adenoids, provides local surveillance where most pathogens first contact you. These stations reflect a design principle: bring rare cells and rare antigens into structured proximity to reduce randomness. Regulation threads through the whole layout. Regulatory T cells, often expressing the transcription factor FOXP3, suppress excessive responses by limiting co-stimulation and emitting calming cytokines. Checkpoint receptors on T cells such as CTLA4 and PD1 dampen activation after initial engagement, akin to speed governors on vehicles. Anti-inflammatory cytokines like IL-10 and TGF-beta reinforce safe braking. Failures in these layers allow autoimmunity, while overzealous breaks permit chronic infection or cancer persistence. Balance, not maximal force, is the aim. Memory rewires the map after a first encounter. Memory B cells and long-lived plasma cells increase antibody quality and availability. Memory T cells patrol tissues and nodes with lower activation thresholds. The next time the city sees the same intruder pattern, responses start earlier and require fewer signals. This economy is why prior exposures, whether through infection or vaccination, shorten the path from recognition to control. Imagine a quiet clinical scene. A person notices painless neck fullness that swells and recedes with colds. On examination, mobile, small lymph nodes suggest reactive hyperplasia, nodes actively filtering and presenting antigens, enlarging as germinal centers form B-cell responses, then regressing. Warning signs such as persistent, hard, fixed enlargement, night sweats, or unexplained fevers warrant professional evaluation because the map can be co-opted by malignancy or chronic infection. This reasoning respects uncertainty while avoiding alarm. You carry an exquisite system that must act decisively without violating home. Understanding its roots lets you interpret the body's signals with steadier confidence. The city lights dim further, and your attention drifts outward from hubs to ramparts. If recognition, regulation, and memory live within, what stands at the very edge to decide what even gets near the gate? Your fingertips meet a fine, slightly acidic film across the skin, a quiet chemistry that greets the world before you do. Barriers are not walls of stone. They are living surfaces with gradients, fluids, and motion. A barrier is any interface that separates internal tissues from the environment while permitting controlled exchange. Microenvironment means the local physical and chemical conditions, pH, oxygen, salt, temperature, moisture, and resident molecules that shape which microbes persist and how cells behave. Together, they form your first lines of protection, deciding what reaches the deeper map. Skin exemplifies layered deterrence. The outer stratum corneum is a compact sheet of dead, flattened keratinocytes embedded in lipids, forming a brick-and-mortar seal that resists water loss and microbial entry. Keratin is a tough structural protein. Its cross-linking stiffens the layer. Intercellular lipids, ceramides, cholesterol, and fatty acids fill gaps to block diffusion. Sebaceous glands deliver sebum, an oily mixture whose breakdown creates an acid mantle with a pH that discourages many pathogens while nurturing selected commensals, microorganisms that live in harmony with you. Tight junctions, protein complexes sealing spaces between living epithelial cells, further restrict paracellular passage below the stratum corneum. When humidity drops or detergents strip lipids, microcracks form and transepidermal water loss rises, subtly weakening defenses and inviting irritants to penetrate. Skin does more than passively block. Keratinocytes sense injury and microbial patterns and release antimicrobial peptides, short, positively charged proteins like defensins and cathelicidins that disrupt microbial membranes. Sweat adds salt and lysozyme, an enzyme that cleaves bacterial cell wall components. Hair follicles and pores are surveilled by resident immune cells, and specialized dendritic cells in the epidermis, often called Langerhans cells, sample antigens and carry messages to lymph nodes. The local microenvironment, slightly acidic, relatively dry, and bathed in low-nutrient sweat, favors commensals that in turn occupy niches, compete for resources, and produce inhibitory metabolites that restrain competitors, a phenomenon termed colonization resistance. Mucosa are moist epithelial linings of the respiratory, gastrointestinal, and urogenital tracts. Their secretions, movement, and resident microbes build dynamic filters where most pathogens first encounter you. In the upper airway, turbinates curve airflow to deposit particles onto mucus, a gel of glycoproteins, mucins that traps microbes. Cilia, microscopic beading projections, move this mucus toward the throat in a coordinated escalator. Coughing and sneezing are reflex expulsions that clear loads when viscosity or particle size overwhelms basal transport. Airway epithelial cells produce antimicrobial peptides, and the lining fluid contains lactoferrin, which binds iron to limit bacterial growth, and secretory immunoglobulin A, secretory IgA, an antibody type adapted to mucosal surfaces. Secretory IgA is an antibody that resists enzymatic degradation and can bind microbes and toxins without provoking strong inflammation. By cross-linking antigens, it prevents adhesion to epithelium, agglutination, and assists ciliary clearance. Surfactant proteins A and D collectins in the lung bind microbial surfaces, enhance phagocytosis, and moderate inflammatory tone. Deeper in the airway, branching geometry increases surface area but narrows passageways. Here, maintaining mucus hydration and ciliary function is critical, which is why high viscosity mucus in certain conditions impairs clearance and predisposes to infection. In the gut, the microenvironment changes sharply along its length. Gastric acid in the stomach creates a very low pH that inactivates many organisms and denatures proteins. In the duodenum, bile salts emulsify fats and disrupt bacterial membranes. Pancreatic enzymes reduce complex carbohydrates and proteins, depriving microbes of macromolecular niches. A rapidly renewing epithelial layer, complete replacement in a matter of days, sheds attached microbes. Mucus forms a two-layer system in the colon, an inner dense layer that limits bacterial access to epithelial cells, and an outer looser layer where microbes reside. Goblet cells produce this mucus. Their activity rises with irritation as a protective surge. Peyer's patches, organized lymphoid structures in the small intestine, lie beneath specialized epithelial cells called M cells that sample luminal contents and deliver them to antigen-presenting cells. This sampling builds tolerance to food antigens while maintaining readiness against pathogens. The gut microbiota, communities of bacteria, archaea, and fungi, metabolize dietary fibers into short-chain fatty acids like butyrate that nourish colonocytes and strengthen tight junctions. They also occupy niches and produce bacteriocins, small proteins that inhibit competitors. Disturbing this community, for example with broad-spectrum antimicrobials, can lower colonization resistance and permit opportunists to bloom. The urogenital tract relies on flow and chemistry. Continuous urine production and voiding mechanically flush microbes. Urothelium expresses glycoprotein receptors in patterns that can either discourage or, if altered, favor bacterial adhesion. In the vagina, lactic acid-producing lactobacilli maintain a low pH that inhibits many pathogens. Hormonal states, hygiene products, and antibiotics can shift this ecology and modulate susceptibility. Tears and saliva contribute persistent washing and specific enzymes. Tears contain lysozyme, lactoferrin, and lipocalin, while blinking spreads a fresh layer that smooths the optical surface and clears debris. Saliva bathes the oral cavity with amylase, mucins, and IgA, supporting a complex oral microbiome that, when balanced, resists invasion. Dental plaque is a biofilm structured microbial communities embedded in a self-produced matrix that adheres to teeth. Biofilms resist penetration by antimicrobials and immune molecules, illustrating how microenvironment and architecture change microbial behavior. Temperature and moisture are quiet regulators. Fever, a regulated rise in body temperature, shifts microbial growth rates and can enhance certain immune processes, but at barriers, even small local temperature changes affect viscosity of mucus and ciliary beat frequency. Moisture gradients influence spore survival and fungal growth. Areas of occlusion on skin, like skin folds, alter pH and humidity, reshaping resident communities and risk profiles. Barriers are adaptive. After injury or microbial sensing, epithelial cells upregulate pattern recognition receptors and secrete cytokines and chemokines, small signaling proteins that attract innate cells. Vascular endothelium in nearby microvessels expresses selectins and adhesion molecules that slow and tether leukocytes. Vascular permeability increases, allowing plasma proteins, including complement components and antibodies, to enter tissues. This permeability carries a trade-off. It delivers resources for defense, but also risks edema and exposure of deeper layers. Resolution mechanisms restore tight junctions, normalize perfusion, and re-establish microbial balance once the threat abates. Consider a calm example. You nick a finger while chopping vegetables. Within minutes, the skin edges weep, clear fluid, as local vessels dilate. Keratinocytes release antimicrobial peptides. Nearby dendritic cells sample the area. If redness spreads rapidly with severe pain out of proportion, or you develop high fever, streaking along the limb, or systemic malaise, those features require timely professional evaluation because they suggest invasion beyond the barrier and possible bloodstream involvement. Most minor cuts close uneventfully as clotting and epithelial migration reseal the surface and the microbiota repopulate their niches. These surfaces negotiate with the world continuously, allowing touch, breath, taste, and intimacy while refusing harm. When they fail, it is often because speed or context shifted, not because their design was simple. You return to the city map and see new lights gather outside the gate. If an intruder crosses the threshold despite mucus, acid, and flow, who arrives first to confront it in the streets? A sudden breach at a cut or in the airway invites the first cellular responders, each arriving with distinct timing and behavior. These are neutrophils, macrophages, and natural killer cells, innate defenders that act before precise memory has time to form. The innate immune system is defined as rapid, non-specific protection. These cells do not rely on prior exposure. Instead, they detect common features of microbes or stress cells and mount immediate responses. Their cooperation is crucial in the window between barrier failure and adaptive activation. Neutrophils are the most abundant circulating leukocytes. They are granulocytes, meaning their cytoplasm contains granules filled with enzymes and antimicrobial compounds. Short-lived, with lifespans measured in hours to a few days, neutrophils act as expendable shock troops. At rest, they patrol the bloodstream. Upon sensing infection signals such as chemokines or complement fragments, they exit vessels by rolling along endothelium, adhering via integrins and squeezing through junctions, a process called diapetesis. Once in tissues, they follow gradients of chemoattractants towards sites of damage or microbial invasion. Their armament is multifaceted. Neutrophils engulf microbes through phagocytosis, enclosing them in phagosomes that fuse with granules containing proteases, defensins, and reactive oxygen species. They can also degranulate, releasing toxic enzymes into the extracellular environment. In addition, neutrophils can expel networks of chromatin and antimicrobial proteins, forming neutrophil extracellular traps, NETs, that immobilize microbes. While effective, this collateral mechanism can damage host tissues and must be tightly regulated. Because neutrophils die quickly, their accumulation forms pus, an opaque fluid of dead cells, bacteria, and debris, marking acute inflammation. Macrophages are longer-lived, versatile cells derived from monocytes that leave the bloodstream and mature in tissues. Unlike neutrophils, many macrophages reside permanently in specific organs, adopting specialized identities: Kupffer cells in the liver, alveolar macrophages in the lungs, microglia in the brain. They act as both sentinels and janitors, phagocytosing debris, apoptotic cells, and pathogens. When they detect microbial or damage signals, macrophages release cytokines such as interleukin-1, tumor necrosis factor alpha, and interleukin-6, which induce fever, recruit neutrophils, and activate endothelium to increase permeability. Macrophages bridge to adaptive immunity by presenting antigens on major histocompatibility complex MHC molecules and providing co-stimulatory signals to T cells. They are also polarized by their environment. Classical activation, sometimes labeled M1, emphasizes microbial killing and inflammation, while alternative activation M2 promotes tissue repair, angiogenesis, and resolution. These are not rigid categories but gradients of function, reflecting how macrophages adapt to context. Their persistence means they influence not only the onset but also the aftermath of immune encounters. Natural killer NK cells are lymphocytes, yet they belong to the innate arm. They lack antigen-specific receptors generated by gene rearrangement, distinguishing them from T and B cells. Instead, NK cells scan for imbalances and signals from activating and inhibitory receptors. Healthy cells display major histocompatibility complex, class I molecules, which provide inhibitory signals to NK cells. Many virus-infected or tumor cells downregulate MHC I to avoid cytotoxic T cells. Paradoxically, this makes them more visible to NK cells, which then receive stronger activating input than inhibitory restraint. Once activated, NK cells release perforin, a protein that forms pores in target cell membranes, and granzymes, enzymes that enter through these pores to trigger apoptosis, programmed cell death. NK cells also secrete interferon gamma, a cytokine that activates macrophages and shapes adaptive responses toward type I helper T cell polarization. Thus, NK cells not only kill directly but also influence the broader immune environment. Their responses are particularly important in early viral infections and in controlling emerging tumors before adaptive cytotoxic T cells expand. The timing of these cells is coordinated. Neutrophils are rapid first arrivals, appearing within hours. Macrophages are already present in tissues and amplify their activity upon stimulation, recruiting neutrophils with cytokines. Monocytes arrive later and mature into macrophages, reinforcing local defense. NK cells typically mobilize within days, overlapping with the handoff to adaptive immunity. This staggered arrival ensures continuous coverage from immediate to sustained responses. Consider an example. A person inhales droplets carrying Streptococcus pneumonia. The bacteria adhere to alveoli, bypassing mucociliary clearance. Alveolar macrophages recognize cell wall components and release cytokines, summoning neutrophils from blood. Within hours, neutrophils flood alveolar spaces, phagocytocyte bacteria, and release reactive oxygen species. Some form NETs that trap clusters of microbes. The battle produces exudate visible on imaging as consolidation. If bacteria breach into blood, NK cells become critical in curbing infected host cells until adaptive immunity generates specific antibodies and cytotoxic T lymphocytes. If instead of resolving, fever, rigors, or confusion develop, those red flags signal the need for professional medical assessment, as sepsis can follow unchecked spread. The actions of these innate cells are effective yet blunt. They often succeed in controlling threats before adaptive responses fully develop. However, their lack of specificity risks collateral tissue damage. Excess neutrophil degranulation erodes tissue. Macrophage cytokine surges can cause systemic inflammation. NK cells can attack transplanted tissues lacking expected self-markers. This balance between speed and precision defines the innate phase. It is reassuring to know your body invests in rapid guardians that sacrifice themselves for immediate protection until the more refined system awakens. The city you envisioned earlier now stirs with patrols and resident guards, but how do these defenders know what to strike and what to spare? The answer lies in the hidden sensors that detect patterns of danger, a story that waits just ahead. The patrols cannot act blindly, they need signals to separate friend from foe. These signals come from molecular sensors embedded in cell membranes or cytoplasm that detect danger patterns. Pattern recognition receptors, abbreviated PRRs, are germline-encoded proteins that recognize conserved molecular motifs. Unlike adaptive receptors, which vary individually, PRRs are fixed blueprints passed through generations. They detect pathogen-associated molecular patterns, PAMPs, and damage-associated molecular patterns, DAMPs. PAMPs are molecular signatures broadly shared by microbes, such as bacterial lipopolysaccharide or viral double-stranded RNA. DMPs are endogenous molecules released from injured or dying cells, such as mitochondrial DNA or high-mobility group box 1 protein. The distinction ensures recognition of both infection and sterile injury. A principal class of PRRs is the toll-like receptors, these TANLARs. These transmembrane proteins are located on plasma membranes or endosomal membranes, positioned to sample extracellular space or ingested material. TR4 recognizes lipopolysaccharide from gram-negative bacteria. TR2 binds lipoteichoic acid from gram-positive bacteria. TR3, 7, 8, and 9 detect nucleic acids, double-stranded RNA, single-stranded RNA, and unmethylated CpG DNA within endosomes, signifying viral or bacterial genomes. Upon ligand binding, TLRs dimerize and trigger intracellular signaling cascades through adapter proteins like MyD88 or TRIF, leading to activation of transcription factors such as NFKB and IRF. These transcription factors induce pro-inflammatory cytokines, type 1 interferons, and costimulatory molecules, setting the stage for both innate defense and adaptive activation. Beyond TLRs, other receptor families expand detection. Node-like receptors, NLRs, reside in the cytoplasm, sensing intracellular bacterial peptidoglycan fragments. Some NLRs assemble into multi-protein complexes called inflammasomes, which activate caspase-1 to cleave pro-IL-1 beta and pro-IL-18 into active cytokines. This initiates strong local inflammation. RIGI-like receptors, RLRs, detect viral RNA in the cytosol, activating interferon responses. C-type lectin receptors, CLRs, bind carbohydrate structures on fungal cell walls. Each family addresses a distinct microbial architecture, ensuring broad coverage. Sensors detect not only foreign patterns but also deviations in context. For instance, extracellular ATP release during necrosis signals danger to purinergic receptors. Oxidized lipids from damaged membranes activate PRRs. Even uric acid crystals can trigger NLRP3 inflammasomes, linking metabolic stress to inflammation. Thus, sensing machinery integrates infectious and non-infectious cues, highlighting immunity's role in tissue homeostasis as well as host defense. Activation of PRRs has immediate consequences. Endothelial cells upregulate adhesion molecules, enabling neutrophil recruitment. Macrophages secrete cytokines that induce fever, mobilize acute phase proteins from the liver, and raise systemic alertness. Dendritic cells upregulate costimulatory molecules and migrate toward lymph nodes, carrying processed antigen fragments. This migration is guided by chemokines and leads to antigen presentation, bridging innate sensing with adaptive activation. The balance is delicate. Overactivation of PRRs can cause harm. Excess TLR signaling may drive septic shock, characterized by systemic vasodilation, hypotension, and multi-organ dysfunction. Chronic inflammasome activity can contribute to autoinflammatory diseases, where tissues are damaged by persistent cytokine release without infection. Conversely, deficient PRR signaling increases susceptibility to infection, as when mutations in TLR pathways predispose individuals to recurrent bacterial or viral illnesses. Recognition must be sensitive yet discriminating. Imagine a scenario: a respiratory virus enters the airway and infects epithelial cells. Viral double-stranded RNA is recognized by TLR3 in endosomes and RIGI in the cytoplasm. These sensors trigger interferon production. Interferons induce an antiviral state in neighboring cells, increasing resistance to viral replication. NK cells are recruited and activated, while dendritic cells capture viral antigens and move to lymph nodes. If instead high fever, confusion, or difficulty breathing arises, professional evaluation becomes necessary, as these may indicate systemic viral illness or pneumonia. Reflection: sensing is perception. Just as your eyes detect patterns of light and your ears patterns of sound, your immune cells detect molecular motifs. They convert perception into action without deliberation. The city you have been picturing now bristles with antennae and detectors, translating molecular whispers into alarms. But alarms alone cannot neutralize intruders. How does the system deploy soluble weapons that attack microbes directly, even before adaptive antibodies appear? That story opens next. From alarms, the city shifts to fountains of cascading reactions that amplify signals into direct attack. This is the complement system, a set of plasma proteins that circulate in inactive forms and become sequentially activated to opsonize pathogens, recruit cells, and directly lyse certain invaders. The word complement reflects its discovery as something that completes the action of antibodies, though it also acts independently. It is part of innate immunity present at baseline and deploys within minutes of encountering microbes. Complement operates through three initiating pathways that converge on common effectors. The classical pathway begins when the C1 complex binds to antibodies already attached to microbial surfaces. This requires adaptive input, but once antibodies exist, complement activation becomes swift. The lectin pathway starts when mannose-binding lectin or phycolins recognize carbohydrate structures on microbial cell walls. The alternative pathway activates spontaneously at low levels, with complement component C3 undergoing hydrolysis. If C3 fragments land on a microbial surface lacking regulators, the pathway amplifies itself. These distinct entry points all funnel toward cleavage of C3. C3 is the central node. Cleavage yields C3A, a small peptide that diffuses as an anaphylatoxin, and C3b, a fragment that covalently attaches to nearby surfaces. C3a induces mast cell degranulation and recruits neutrophils, enhancing inflammation. C3b coats pathogens, making them recognizable to phagocytes bearing complement receptors, a process called opsonization. This tagging greatly improves efficiency of engulfment, especially for microbes resistant to direct killing. C5 is another key component. C3b contributes to forming C5 convertase, which cleaves C5 into C5a and C5b. C5a is among the most potent chemoattractants, drawing neutrophils and monocytes and amplifying vascular permeability. C5b initiates assembly of the membrane attack complex MAC by binding C6, C7, C8, and multiple C9 molecules. Together they form a pore that inserts into microbial membranes, leading to osmotic lysis. This is particularly effective against gram-negative bacteria. The system is tightly regulated to protect host cells. Regulators such as decay-accelerating factor DF disrupt convertices, factor H directs inactivation of C3b on host membranes, and CD59 prevents MAC formation on self cells. Without these checks, complement could damage host tissues. Deficiencies in regulators lead to conditions such as paroxysmal nocturnal hemoglobinuria, where red blood cells are lysed by uncontrolled complement. Conversely, deficiencies in components predispose to recurrent infections, especially with Neisseria species when terminal pathway proteins are absent. Complement does not act in isolation. Its fragments shape adaptive immunity. C3d, a breakdown product of C3b, binds to B-cell receptors along with antigen, lowering the threshold for B-cell activation. This illustrates how soluble innate cascades prepare adaptive cells to respond with speed and sensitivity. Complement also cooperates with antibodies in clearance of immune complexes, reducing deposition in tissues and lowering risk of inflammatory injury. Consider a quiet clinical picture. A child presents with repeated meningococcal infections despite vaccination. Investigation reveals deficiency in a late complement component. The absence of MAC formation prevents efficient killing of Neisseria. Explaining susceptibility. Management involves preventive strategies and professional monitoring. This vignette highlights how identifying missing parts clarifies function. Complement activation illustrates the design of cascades. Each step amplifies the signal, much like a series of dominoes that topple with increasing force. Small initiating events yield large responses, ensuring rapid expansion when a pathogen appears, yet amplification without control risks systemic harm. Excessive complement activation contributes to sepsis, where widespread vascular leakage and inflammation endanger organs. Thus, regulators and termination mechanisms are as vital as effectors. Brief reflection. Cascades remind you of precision hidden in your blood, silent molecules that can explode into action within moments, then vanish again when no longer needed. As you look over the city, you notice that alarms and cascades have summoned reinforcements. The air grows warm, vessels widen, and tissue swells. What orchestration makes redness, heat, and pain appear? And how does the system know when to stop? That is the next chapter. You feel warmth spread under the skin around a small wound, as if the tissue itself were exhaling heat. This is inflammation, a carefully orchestrated process that protects and repairs. Inflammation is defined as a coordinated vascular and cellular response to injury or infection, characterized by redness, heat, swelling, and pain. These signs arise from underlying mechanisms that ensure immune cells and plasma proteins reach the site efficiently. The sequence begins with local recognition of danger. Macrophages, dendritic cells, and mast cells detect pathogens or damage-associated molecules and release mediators such as histamine, prostaglandins, leukotrienes, and cytokines. Histamine, stored in mast cell granules, rapidly dilates nearby arterioles and increases vascular permeability. Prostaglandins and leukotrienes, derived from arachidonic acid in membranes, prolong dilation and promote leukocyte recruitment. Cytokines like tumor necrosis factor alpha and interleukin-1 activate endothelium, causing it to express adhesion molecules. Vasodilation explains redness, rubor, and heat. Calor, increased blood flow brings warmth and color to the tissue. Increased vascular permeability allows plasma proteins and fluid to exit into interstitial spaces, producing swelling, tumor. Pain dolor arises from direct mediator effects on sensory nerve endings, particularly bradykinin and prostaglandins, as well as pressure from edema. Together, these changes produce loss of function, functionalasa, a classic addition to the 4 cardinal signs. Leukocyte recruitment follows a multi-step adhesion cascade. Circulating neutrophils slow down by binding selectins, roll along the endothelium, then activate integrins that mediate firm adhesion. They squeeze through endothelial junctions and migrate along chemokine gradients into tissue. Monocytes and later lymphocytes follow. Complement fragments C3a and C5a, bacterial peptides and chemokines like IL-8 act as attractants. This migration ensures that the site of injury fills with effector cells able to phagocytose microbes and secrete further mediators. Mediators are numerous but organized. Interleukin-6 stimulates liver synthesis of acute phase proteins like C-reactive protein, which opsonizes microbes and activates complement. Tumor necrosis factor increases vascular permeability systemically and can induce fever by acting on the hypothalamus. Interleukin-1 reinforces this pyrogenic signal and enhances lymphocyte activation. Together, these molecules raise systemic readiness while local effects continue. Resolution is as important as initiation. Anti-inflammatory mediators like interleukin-10 and transforming growth factor beta restrain responses. Lipid mediators switch from pro-inflammatory leukotrienes to pro-resolving lipoxins, resolvins, and protectins. These molecules promote clearance of neutrophils by macrophages, encourage tissue repair, and restore vascular integrity. Failure of resolution risks chronic inflammation, where persistent mediators and cell infiltration cause tissue damage and fibrosis. Inflammation illustrates cooperation across systems. The nervous system transmits pain and regulates vascular tone. The vascular system delivers cells and proteins. The immune system directs the choreography. The process protects against microbes and clears debris, but excessive or misdirected inflammation causes harm. In sepsis, overwhelming mediator release causes systemic vasodilation and capillary leakage, threatening circulation. In autoimmune conditions, chronic inflammation damages tissues despite absence of pathogens. Imagine a clinical vignette. A person develops a red, hot, swollen joint after a minor cut. Local inflammatory signs indicate immune activation and tissue repair. If swelling becomes rapidly progressive with severe pain, fever, or inability to bear weight, professional evaluation is essential, as infection within a joint can cause permanent damage. Most minor inflammatory responses, however, subside as resolution mediators restore balance. Reflection: inflammation is the language of alarm, loud, visible, and tangible. Yet within its heat lies a script that ends with calm if allowed to complete. The city you picture glows now with widened vessels and swelling streets, but the question remains, how does the system communicate the details of what has invaded, handing fragments from one arm to the other? That is where antigen presentation begins. A courier enters the city square carrying fragments of an intruder displayed like badges on a board. This is antigen presentation, the process by which immune cells display molecular pieces of microbes or abnormal cells to lymphocytes, linking innate recognition to adaptive precision. Without this handoff, adaptive immunity cannot be properly activated. Antigen is any molecule that can be recognized by the receptors of B or T lymphocytes. For T cells in particular, recognition requires antigens to be processed into peptides and presented on specialized molecules called major histocompatibility complex, MHC. There are two primary classes of MHC, each routing information from a distinct source. MHC class I is expressed on nearly all nucleated cells. It samples intracellular proteins by binding peptides generated from proteasomal degradation. Transporter proteins called TAP carry these peptides into the endoplasmic reticulum where they are loaded onto MHC I. The complex then travels to the cell surface. Infected or abnormal cells thus present internal fragments. Cytotoxic CD8 T cells scan MHC I, and if foreign or altered peptides are detected, they trigger targeted killing. This pathway guards against viruses and malignant transformation. MHC class II is expressed mainly on professional antigen-presenting cells, dendritic cells, macrophages, and B cells. These cells internalize extracellular material through phagocytosis or endocytosis, degrade it within acidified compartments, and load resulting peptides onto MHC II molecules. Once at the surface, MHC II presents antigens to CD4 helper T cells. Recognition by CD4 T cells provides the help required for antibody production, macrophage activation, and orchestration of adaptive responses. Co-stimulation is essential. A T cell receptor binding peptide MHC alone is not sufficient. A second signal is needed to prevent inappropriate activation. Antigen-presenting cells provide this by expressing molecules such as CD80 and CD86, which engage CD28 on T cells. Only in the presence of both signals, often induced by innate sensing of danger, do T cells activate. Absence of co-stimulation leads to anergy, a state of functional silence that enforces tolerance. Dendritic cells are the archetypal professional presenters. They reside in peripheral tissues in an immature state, highly endocytic but not yet stimulatory. Upon encountering pathogen-associated signals, they mature soon, upregulating MHC, co-stimulatory molecules, and cytokine production. They migrate via lymphatic vessels to regional lymph nodes, guided by chemokines. There, in the T cell zones, they extend processes to interact with naive T cells, providing antigen, co-stimulation, and polarizing cytokines. This is the critical junction where innate sensing directs adaptive differentiation. B cells also act as presenters, though with a twist. Their surface immunoglobulin acts as an antigen receptor, binding a specific molecular structure. After internalizing this bound antigen, B cells present peptide fragments on MHC II to helper T cells. In return, T cells provide signals that drive B cell activation, proliferation, and antibody production. This reciprocal presentation builds specificity and memory. Antigen presentation also involves cross-presentation, a process where dendritic cells ingest extracellular antigens and present them on MHC I, thereby activating CD8 cytotoxic T cells. This is crucial for initiating responses against viruses that do not directly infect dendritic cells and against tumors. Failures in antigen presentation carry consequences. Defects in MHC molecules or peptide transport can cause immunodeficiency as T cells fail to recognize threats. Conversely, aberrant presentation of self-peptides without proper tolerance mechanisms contributes to autoimmunity. Pathogens can subvert presentation. Some viruses downregulate MHC I to avoid CD8 T cells, while others interfere with peptide loading. Yet these evasions often increase vulnerability to NK cell surveillance. Consider a vignette. A person contracts influenza. Infected epithelial cells present viral peptides on MHC I, drawing cytotoxic T cells. Meanwhile, dendritic cells ingest viral debris, migrate to lymph nodes, and present peptides on MHC II to helper T cells. These T cells, once activated, provide signals that enable B cells to produce high-affinity antibodies. If instead someone shows prolonged fever, severe shortness of breath, or confusion, These signs warrant professional care, as they may signal severe or systemic infection. Reflection, antigen presentation is conversation. Cells displaying fragments of their experience, others listening and deciding action. It is language written in proteins, read with precision. In the city you have been following, the couriers have spoken. Now the crowd of helper T cells must decide roles, splitting into specialized voices that shape the coming response. Which voices dominate, and what do they ask the system to do next? A gathering of helper T cells forms around the presented antigens, each cell leaning toward a different path of instruction. These are CD4 T Lymphocytes, central coordinators that decide whether to inflame, calm, or specialize the immune response. Their differentiation is guided by the cytokine environment at the moment of activation, shaping them into distinct subsets with tailored functions. Thymus cells emerge when dendritic cells and macrophages secrete interleukin-12 and interferon gamma. Thymus cells in turn secrete more interferon gamma, creating a reinforcing loop. Their role is to activate macrophages into heightened antimicrobial states and to support cytotoxic T lymphocyte development. Thymus dominance is crucial against intracellular bacteria such as Mycobacterium tuberculosis and certain viruses. Overactivity, however, can drive tissue-damaging inflammation. D2 cells differentiate in the presence of interleukin-4. They secrete interleukin-4, interleukin-5, and interleukin-13, promoting B-cell class switching to IgE and recruiting eosinophils and basophils. This pathway excels at combating helminths and other multicellular parasites. Yet when misapplied, D2 responses underlie allergic conditions like asthma and atopic dermatitis, where IgE-mediated reactions misinterpret harmless antigens as threats. Th17 cells arise under the influence of interleukin-6, interleukin-23, and transforming growth factor beta. They secrete interleukin-17 and interleukin-22, recruiting neutrophils and fortifying epithelial barriers. These cells are critical against extracellular bacteria and fungi at mucosal surfaces. Dysregulated Th17 activity contributes to autoimmune diseases such as psoriasis and inflammatory bowel disease, where neutrophil recruitment damages host tissue. Regulatory T cells, often marked by expression of the transcription factor FOXP3, differentiate when transforming growth factor beta dominates without strong inflammatory cues. They secrete interleukin-10 and transforming growth factor beta, dampening immune responses and maintaining tolerance. Without them, unchecked activation risks autoimmunity. Too many, conversely, may permit persistent infection or tumor escape. The balance among TA1, TA2, TA17, and regulatory subsets defines immune outcomes. In leprosy, for example, tuberculoid forms show strong TA1 responses with controlled bacterial load, while lepromatous forms show TA2 bias with disseminated disease. In asthma, excessive TA2 activity drives airway inflammation, while in chronic fungal infection, insufficient TA17 activity weakens mucosal defense. These clinical contrasts reveal how helper T cell polarization determines not only whether immunity succeeds but also how it might harm. CD4 T cells act not in isolation but in conversation. Through CD4T, CD40L interactions, they deliver activating signals to B cells and macrophages. Through cytokine gradients, they influence local cells, endothelial cells, fibroblasts, and epithelial cells to modulate permeability and repair. Their presence or absence dictates the quality of adaptive responses. Without helper input, cytotoxic T cells may fail to persist and B cells may fail to generate high-affinity antibodies. Consider a scenario. A patient exposed to Candida albicans at mucosal surfaces requires Th17-driven neutrophil recruitment for clearance. If this pathway is defective, recurrent oral or genital candidiasis occurs. Recognition of such patterns signals the need for professional evaluation. In contrast, robust Th17 responses normally confine fungi and restore barrier function. The clinical outcome reflects the tuning of helper T cells. Reflection: within one cell type lies a chorus of voices, some urging attack, some calling for restraint. Your immune system thrives not on a single command, but on harmony among many. In the city analogy, the assembly has spoken, assigning leaders to mobilize, recruit, or calm. Yet another group waits nearby, the cytotoxic enforcers who eliminate compromised cells and then quietly leave behind long-lived guardians. Who are these executors, and how do they remember? From the gathering of helpers steps a cadre of quieter, more decisive figures, CD4 T Lymphocytes, often called cytotoxic T cells. Their role is direct elimination of cells harboring threats. Unlike helper T cells, which orchestrate, CD8 cells execute. They recognize peptides displayed by major histocompatibility complex class I, MHC I, which appears on nearly all nucleated cells. Thus, any cell that becomes infected with a virus or undergoes malignant transformation can present abnormal peptides on MHC I and become a target. Activation of CD8 T cells requires two key inputs: recognition of antigen MHC I complexes and co-stimulation provided by antigen-presenting cells. Dendritic cells often achieve this through cross-presentation, displaying exogenous antigens on MHC I to prime naive CD8 T cells. Helper T cells add another layer of support by secreting cytokines like interleukin 2, which fuels proliferation. Once fully activated, CD8 T cells expand into large clonal populations of effector cells, each tuned to a specific peptide signature. Their weapons are precise. Perforin creates pores in target cell membranes, through which granzymes enter to trigger apoptosis. This programs cell death, dismantles intracellular pathogens without releasing their components chaotically. CD8 T cells also express Fas ligand, engaging Fas receptors on target cells to initiate apoptosis through a separate pathway. These redundant mechanisms ensure efficient clearance of infected or transformed cells. The expansion phase is intense. Within days of activation, clonal numbers of CD8 T cells can increase dramatically, swarming through blood and tissues. Their presence is transient. Once the threat is cleared, most undergo apoptosis in a contraction phase, reducing risk of unnecessary tissue injury. Only a small proportion persist as memory CD8 T cells poised for rapid response upon re-exposure. Memory CD8 T cells are specialized in their longevity and readiness. Central memory T cells recirculate through lymphoid organs, while effector memory T cells patrol peripheral tissues. Tissue-resident memory T cells remain embedded in local barriers like the skin or gut, forming rapid response stations. These populations enable swift elimination of infected cells at future encounters, shortening the delay between recognition and action. Clinical relevance underscores their importance. Viral infections such as influenza or hepatitis rely heavily on cytotoxic T cells for resolution. In HIV, progressive loss of CD4 help impairs CD8 function, leading to vulnerability. In oncology, CD8 responses can initially control tumor growth, but tumors may evade detection by downregulating MHC I or secreting inhibitory molecules. Therapies such as checkpoint inhibitors aim to restore CD8 vigor by lifting inhibitory brakes. Consider an example. A person develops shingles caused by reactivation of varicella-zoster virus in nerve ganglia. Infected neurons present viral peptides on MHC I. CD8 T cells recognize these peptides and induce apoptosis, containing viral spread along dermatomes. If instead the reactivation spreads unchecked with disseminated rash and systemic symptoms, professional medical care becomes essential, as this reflects failure of containment. Reflection. Cytotoxic T cells are silent surgeons, excising threats one cell at a time, then fading back into memory. Their strength lies in restraint as much as in execution. Within your imagined city, these enforcers carry out precise removals, leaving behind only trusted guards. Yet across the plaza, another lineage prepares a different strategy, the secretion of antibodies that circulate like passports and traps. Who trains these cells, and how do their secretions shape the flow of defense? Across the city map, another lineage takes shape, B lymphocytes, the architects of antibody production. Where CD8 cells eliminate infected cells directly, B cells release soluble molecules that circulate broadly. Antibodies, also called immunoglobulins, bind antigens with high specificity, marking them for clearance, neutralizing toxins, and preventing microbial attachment. B cells begin as naive cells in bone marrow, each bearing a unique surface immunoglobulin receptor generated by gene rearrangement. When this receptor encounters its matching antigen and receives help from CD4 T cells, the B cell becomes activated. Activation drives clonal expansion and differentiation into two major fates: plasma cells, which secrete antibodies in high quantity, and memory B cells, which persist for rapid recall. Antibodies exist in several isotypes, each with distinct properties. AGM is the first produced. It forms pentamers, providing strong agglutination capacity, though with relatively low affinity. AGG is the most abundant in blood and extracellular fluid, versatile in neutralization, opsonization, complement activation, and neonatal transfer across the placenta. AGA predominates at mucosal surfaces, often existing as a dimer secreted into saliva, tears, breast milk, and intestinal secretions, providing frontline defense without provoking strong inflammation. AGE is the least abundant, but binds tightly to mast cells and basophils. Upon cross-linking by antigen, it triggers release of histamine and other mediators, important in defense against helminths, but also central to allergic responses. AGD, less well understood, resides mainly as a receptor on naive B cells. Affinity maturation refines antibody quality. Within germinal centers of lymph nodes, B cells undergo somatic hypermutation, small changes in their antibody genes. Those producing higher affinity antibodies receive survival signals from follicular helper T cells. Over time, the antibody pool improves in binding strength, a process called affinity maturation. Alongside, class switching replaces the constant region of the antibody gene, enabling transition from IgM to IgG, IgA, or IgE, while retaining specificity. This tailoring ensures antibodies match the site and nature of the threat. Plasma cells are specialized factories. Short-lived plasma cells produce bursts of antibody early, while long-lived plasma cells reside in bone marrow, secreting antibodies for months to years, sustaining protection after infection or vaccination. Memory B cells do not secrete antibody until re-stimulated, but upon re-exposure, they expand rapidly, accelerating and magnifying responses. This explains why second encounters with pathogens are often mild or asymptomatic. Antibodies act through several mechanisms. Neutralization involves binding to microbial proteins or toxins, preventing their interaction with host receptors. Opsonization coats pathogens with antibodies, facilitating recognition by phagocytes through Fc receptors. Complement activation occurs when antibody-antigen complexes trigger the classical pathway, enhancing opsonization and lysis. Antibody-dependent cellular cytotoxicity recruits NK cells to kill targets coated with IgG. Each mechanism extends the reach of adaptive immunity into extracellular spaces. Clinical patterns highlight their significance. Deficiency in B cells or antibodies predisposes to recurrent bacterial infections, particularly with encapsulated organisms like Streptococcus pneumoniae. In contrast, excessive IgE production drives allergic disease. Therapeutic antibodies engineered in laboratories now target tumors, inflammatory mediators, or viral proteins, harnessing antibody biology for treatment. Consider a vignette. An infant born with impaired B-cell development presents with recurrent ear infections and pneumonia. Absence of circulating immunoglobulins explains vulnerability. Professional assessment confirms immunodeficiency, requiring supportive strategies. In contrast, in a vaccinated child, robust IgG responses prevent illness upon pathogen exposure, illustrating how antibodies sustain long-term protection. Reflection: antibodies are notes written by your immune history, circulating silently, ready to block what once caused harm. They are memory-made fluid. In your cityscape, antibodies spread like signals along trade routes, guarding at distance. Yet these signals need organized stations, organs where training, activation, and circulation converge. What structures provide these meeting places, and how does lymph know where to travel? The city cannot function without central stations and checkpoints where cells are born, trained, and routed. These are the lymphoid organs, primary sites for development and secondary sites for activation and coordination. Together they shape how lymphocytes circulate and where immune encounters occur. Primary lymphoid organs are bone marrow and thymus. Bone marrow, nestled within bones, is where hematopoietic stem cells generate all blood lineages. B cells mature here, undergoing gene rearrangement to produce unique receptors. Immature B cells that bind strongly to self-antigens are deleted or edited, a process of central tolerance. The thymus, located above the heart, educates T cells. Precursors arriving from marrow rearrange their receptors, then undergo positive selection to ensure they recognize self-MHC, followed by negative selection to eliminate strongly self-reactive clones. The thymic cortex and medulla, with epithelial and dendritic cells, orchestrate this rigorous training. By young adulthood, thymic tissue gradually involutes, but its early imprint persists in long-lived T cells. Secondary lymphoid organs include lymph nodes, spleen, and mucosa-associated lymphoid tissue, MLT. Lymph nodes are small, bean-shaped structures distributed along lymphatic vessels. Lymph, a fluid derived from interstitial spaces, carries antigens and dendritic cells into nodes. Within nodes, B cells localize to follicles, and T cells to paracortical areas, optimizing interactions. Follicular dendritic cells present intact antigens to B cells, while conventional dendritic cells present processed peptides to T cells. This architecture maximizes chance meetings between rare lymphocytes and their cognate antigens. The spleen is the major filter of blood. Its white pulp houses lymphocytes arranged around arterioles, while red pulp removes aged red cells and pathogens. Unlike lymph nodes, which sample lymph draining from tissues, the spleen surveys antigens circulating in blood. This distinction explains why loss of spleen function predisposes to infections with encapsulated bacteria. Without splenic macrophages and marginal zone B cells, rapid antibody responses are impaired. MALT represents diffuse surveillance at mucosal surfaces, entry points for most pathogens. Examples include gut-associated lymphoid tissue, GALT, such as Peyer's patches, bronchus-associated lymphoid tissue, B-BALT, and structures in the nasopharynx like tonsils and adenoids. Specialized epithelial cells called M cells transport antigens from the lumen to underlying immune cells. This enables rapid responses while maintaining tolerance to food and commensals. The balance here is delicate. Exaggerated responses produce food allergies or inflammatory bowel disease, while insufficient responses permit chronic infection. Cell trafficking relies on chemokines and adhesion molecules. Naive lymphocytes enter nodes from blood via high endothelial venules directed by selectins and chemokine receptors. If they do not encounter their specific antigen, they exit via efferent lymphatics to continue circulation. This constant recirculation ensures broad surveillance by a limited pool of lymphocytes. Once activated, lymphocytes alter their homing receptors to migrate preferentially to inflamed tissues or mucosal surfaces, depending on where activation occurred. Clinical examples underscore organ roles. Splenectomy after trauma or disease removal increases risk of sepsis from encapsulated organisms, requiring preventive care. Lymphadenopathy and large nodes may reflect reactive proliferation during infection, but persistent hard or fixed nodes require professional evaluation to exclude malignancy. Thymic defects, such as in DiGeorge syndrome, cause profound T-cell deficiency, leading to recurrent infections. These scenarios highlight how organ integrity underpins immune readiness. Reflection: lymphoid organs are less visible than beating hearts or breathing lungs. Yet they are the classrooms and marketplaces of immunity, where knowledge and defense meet. In your imagined city, stations hum with activity, guiding traffic of lymphocytes through nodes, spleen, and mucosa. Yet even in this order, conversations rely on chemical messengers. What words do immune cells speak to one another, and which voices dominate when the system is stirred? The city's guards and couriers cannot coordinate without shared language. That language is written in cytokines, small proteins secreted by immune and non-immune cells that act as messengers. A cytokine is defined as a signaling molecule that binds to specific receptors on target cells, altering their behavior. Cytokines regulate intensity, duration, and quality of immune responses, functioning locally or systemically. Several cytokines are central to early defense. Interleukin-1 IL-1 is produced by activated macrophages and dendritic cells. It induces fever by acting on the hypothalamus, increases adhesion molecules on endothelial cells to facilitate leukocyte recruitment, and stimulates production of acute phase proteins in the liver. Interleukin-6 IL-6, also secreted by macrophages and endothelial cells, amplifies acute phase responses, promotes B-cell differentiation, and contributes to fever. Tumor necrosis factor alpha, TNF-alpha, secreted mainly by macrophages, is a potent mediator of vascular permeability, leukocyte recruitment, and systemic inflammation. Interferons, particularly type 1 interferons, IFN-alpha and IFN-beta, are produced by virus-infected cells and plasmacytoid dendritic cells. They induce an antiviral state in neighboring cells, upregulate antigen presentation, and recruit natural killer cells. These cytokines rarely act alone. They form networks of synergy and antagonism. IL-1 and TNF-alpha reinforce each other in promoting endothelial activation. IL-6 complements their action by shaping acute phase responses. Type 1 interferons cross-prime adaptive responses by stimulating dendritic cells and enhancing cytotoxic T-cell activity. Regulatory cytokines like IL-10 and transforming growth factor beta counterbalance these effects, ensuring inflammation resolves. A dramatic manifestation of unrestrained cytokine release is the cytokine storm. This term describes excessive, systemic production of pro-inflammatory cytokines leading to widespread vascular leakage, coagulation abnormalities, and multi-organ dysfunction. Cytokine storms can occur in severe viral infections, certain autoimmune diseases, or as complications of immunotherapies such as CAR-T cells. Clinically, they present with high fever, hypotension, respiratory distress, and coagulopathy. Their existence illustrates that the same molecules protecting against pathogens can, in excess, threaten survival. Cytokines also specialize adaptive responses. Interleukin-12 drives differentiation of helper T cells toward the Thy1 lineage, supporting macrophage activation. Interleukin-4 fosters Thy2 development and IgE production. Interleukin-23 supports Thy117 expansion, enhancing neutrophil recruitment. These polarizing cytokines determine whether the immune system leans toward intracellular defense, extracellular defense, or regulation. Consider an example. During bacterial pneumonia, alveolar macrophages release IL-1, IL-6, and TNF-alpha, producing fever and acute phase proteins. These cytokines recruit neutrophils into alveoli, where they phagocytocyte bacteria. If systemic symptoms escalate with confusion or severe hypoxemia, this indicates excessive mediator activity and requires professional care. Normally, once infection resolves, IL-10 and TGF-beta predominate, dampening responses and promoting tissue repair. Reflection. Cytokines are voices, not weapons. They persuade, summon, and calm, shaping how immune actors behave. In their balance lies the difference between healing and harm. The city you have been following now vibrates with these spoken signals. Some voices shout danger, others whisper restraint, but sometimes restraint fails at its deepest level. How does the immune system normally avoid harming its own body, and what happens when tolerance breaks? In the city you have been tracing, the greatest challenge is not the invader, but the risk of turning weapons inward. Tolerance is the system's commitment to spare self, achieved through layered safeguards. When tolerance falters, autoimmunity emerges, conditions where the immune system mistakenly attacks the body's own tissues. Central tolerance occurs during lymphocyte development. In the thymus, developing T cells undergo positive selection to ensure they recognize self-MHC, then negative selection to eliminate those binding too strongly to self-peptides. Medullary thymic epithelial cells express a wide range of tissue-specific antigens under the control of the ARI gene, exposing T cells to otherwise hidden proteins. In bone marrow, developing B cells that strongly bind self-antigen may undergo receptor editing, deletion, or anergy induction. These central processes reduce but do not eliminate autoreactive cells. Peripheral tolerance addresses those that escape. Regulatory T cells suppress activation by releasing inhibitory cytokines and by direct contact-dependent mechanisms. Allergy arises when lymphocytes encounter antigen without co-stimulation, leaving them functionally silent. Apoptosis can follow chronic, repeated activation without survival signals. Checkpoint molecules like CTLA-4 and PD-1 further restrain T-cell activity by competing with activating receptors. Together, these mechanisms form a net of restraint outside primary organs. When these safeguards fail, autoimmunity develops. Systemic lupus erythematosus (SLE) illustrates loss of tolerance to nuclear antigens, producing autoantibodies against DNA and ribonucleoproteins. These form immune complexes that deposit in tissues, causing nephritis, arthritis, and rashes. Rheumatoid arthritis reflects autoreactivity to synovial components, driving chronic joint inflammation and destruction. Graves' disease arises when autoantibodies stimulate the thyroid-stimulating hormone receptor, leading to overproduction of thyroid hormones. Each condition shows how specific breaches in tolerance manifest as distinct syndromes. Genetic and environmental factors both contribute. Certain HLA alleles increase susceptibility, suggesting that particular antigen-presenting frameworks influence self-recognition. Infections can provide danger signals that break anergy or molecular mimicry, where microbial antigens resemble self-antigens, tricking the system into cross-reactivity. Tissue damage releasing sequestered antigens, such as after trauma, can also provoke autoreactivity. Regulation remains possible even after breaches. Many autoimmune conditions wax and wane, reflecting ongoing tension between effector and regulatory pathways. Therapies often aim to restore balance by dampening effector activity or boosting regulatory mechanisms. Consider a vignette. A young adult presents with joint pain, morning stiffness, and swollen fingers. Laboratory studies reveal autoantibodies and inflammatory markers. While this constellation may suggest autoimmune disease, professional evaluation is crucial, as diagnosis and management depend on comprehensive assessment. For most people, transient aches resolve and tolerance holds firm, but persistence, systemic patterns require expert guidance. Reflection. Tolerance is restraint in the midst of power. To know self and yet hold back is as important as striking down what does not belong. In your imagined city, restraint has bent but not always held. When tolerance breaks completely, the immune system can not only attack itself but also react too strongly to harmless outsiders. How does it misinterpret allergens or mount exaggerated reactions against otherwise minor triggers? You notice in the city that not all alarms are directed against dangerous invaders. Sometimes the system reacts to harmless dust, foods, or its own tissues in ways that resemble misfired drills. These exaggerated or misdirected responses are called hypersensitivities, divided into 4 broad types based on mechanism and timing. Type I hypersensitivity is immediate and IgE-mediated. On first exposure to an allergen, B cells class switch to IgE under the influence of Th2 cells and interleukin-4. IgE binds tightly to mast cells and basophils. Upon re-exposure, allergens cross-link IgE, triggering degranulation of histamine, leukotrienes, and prostaglandins within minutes. The result is urticaria, rhinitis, bronchospasm, or even anaphylaxis, widespread vasodilation, airway narrowing, and hypotension. This reaction illustrates defense machinery against helminths misapplied to pollen or peanuts. Type 2 hypersensitivity involves IgG or IgM antibodies directed against antigens on cell surfaces or extracellular matrix. Binding of antibody activates complement or recruits effector cells, leading to cytotoxicity. Classic examples include autoimmune hemolytic anemia, where red cells are destroyed, and Graves' disease, where stimulatory autoantibodies target the thyroid receptor. These reactions are generally localized to specific tissues bearing the targeted antigen. Type 3 hypersensitivity results from immune complexes, antigen-antibody aggregates that deposit in tissues and activate complement. The resulting inflammation damages vessels and organs. Serum sickness is an example, historically seen when foreign proteins were injected for therapy. Systemic lupus erythematosus also demonstrates this mechanism, with immune complexes depositing in kidneys, joints, and skin. The common thread is injury not from direct antibody binding but from accumulation of complexes. Type 4 hypersensitivity is delayed and T-cell mediated. Unlike antibody-driven types I-III, Type 4 involves sensitized T cells that recognize antigen presented by MHC. Upon re-exposure, they release cytokines that recruit macrophages and other effectors, with responses appearing after 24 to 72 hours. Contact dermatitis from nickel exposure, the tuberculin skin test, and tissue damage in chronic infections like tuberculosis illustrate this mechanism. Because it relies on cellular recruitment and activation, The onset is slower but potentially persistent. All 4 hypersensitivity types reflect normal defense strategies: rapid IgE reactions against parasites, cytotoxic antibodies against infected cells, immune complex clearance, and T-cell mediated microbial control, misapplied or exaggerated. The same architecture that protects can harm when regulation fails or targets are misidentified. Consider a clinical vignette. A person eats shellfish and develops sudden lip swelling, wheezing, and lightheadedness. These symptoms suggest systemic IgE-mediated reaction. Immediate professional care is vital in such scenarios, as airway compromise and shock can follow. In contrast, mild seasonal rhinitis reflects localized type 1 hypersensitivity and usually resolves after exposure ends. The spectrum underscores the importance of recognizing severity. Reflection: hypersensitivity is the cost of a vigilant system. It errs sometimes on the side of overreaction, mistaking a breeze for a storm. In the city you have been following, false alarms have sounded, some minor, some dangerous. But what if instead the walls themselves are thin and defenders are missing or suppressed? That is the reality of immunodeficiency. In the city you have been picturing, sometimes the guards are not overzealous but scarce, absent, or weakened. This state is called immunodeficiency, defined as impaired function of one or more components of the immune system, leading to increased susceptibility to infection. Immunodeficiencies can be primary, arising from genetic defects, or secondary, acquired through external factors. Primary immunodeficiencies are usually evident in childhood. Severe combined immunodeficiency (SCID) exemplifies a profound defect. Both T and B cells are absent or non-functional. Infants with SCID develop recurrent, severe infections from bacteria, viruses, and fungi. Without intervention, survival is limited, underscoring the necessity of functional adaptive immunity. Another example is common variable immunodeficiency (CID), where B-cell differentiation into plasma cells is impaired, leading to low antibody levels. Patients present with recurrent respiratory and gastrointestinal infections. Chronic granulomatous disease, in contrast, reflects defective neutrophil oxidative burst, leaving phagocytes unable to kill certain catalase-positive bacteria and fungi. Each disorder highlights how loss of a single mechanism unmasks the importance of that pathway. Secondary immunodeficiencies are more common. Human immunodeficiency virus, HIV, targets CD4 T cells, leading to progressive depletion. Without helper cells, cytotoxic T and B cell responses falter, leaving the host vulnerable to opportunistic infections and certain cancers. Medications such as corticosteroids suppress broad immune functions, dampening inflammation but also reducing defense against microbes. Chemotherapy, malnutrition, advanced age, and chronic diseases can also weaken immunity. Unlike primary forms, secondary immunodeficiencies can arise at any point in life. Patterns of infection often point toward the underlying deficit. Recurrent bacterial sinus and lung infections suggest antibody deficiencies. Opportunistic viral or fungal infections suggest T-cell defects. Abscesses with unusual organisms may indicate neutrophil dysfunction. Systemic invasive infections with encapsulated bacteria are typical in complement deficiencies. Recognition of these patterns helps professionals guide further evaluation. The balance between deficiency and excess is delicate. Immunosuppression may be intentional, for example, after organ transplantation to prevent rejection, yet the cost is increased vulnerability. Conversely, failure to suppress after intended therapy can leave patients at risk of autoimmune complications. The spectrum shows that immune strength is not a simple scale, but a tailored calibration. Consider a vignette. A young child presents with recurrent pneumonia, thrush, and chronic diarrhea. Concern arises for profound immunodeficiency, and urgent professional evaluation is essential. In contrast, an adult with transient neutropenia after chemotherapy may experience mouth ulcers and fever that resolve as counts recover, illustrating how temporary deficiencies also shift vulnerability. Reflection: the immune system is not invincible armor, but a living balance. Its absence reveals fragility, while its restoration renews resilience. In your imagined city, you have seen alarms, excesses, and absences, But can defenses be trained deliberately so that memory arises without disease? That is the principle of vaccination. The next chapter. Imagine preparing the city for an attack before any invader arrives. This is the principle of vaccination, deliberate exposure to a safe form of antigen that induces immune memory without causing disease. Vaccines are among the most powerful public health interventions, training adaptive immunity to respond quickly and effectively at real encounters. Several categories of vaccines exist. Live-attenuated vaccines use pathogens weakened so they replicate poorly in humans, but still elicit strong immune responses. Because they mimic natural infection, they often induce durable cellular and humoral immunity. Examples include vaccines against measles and yellow fever. Inactivated vaccines use killed organisms, unable to replicate but still presenting antigenic structures. These are safer for immunocompromised individuals, but generally require boosters to sustain protection. Subunit vaccines contain purified components, proteins or polysaccharides from pathogens. They are highly safe and specific but may need adjuvants to enhance immunogenicity. Recent advances include mRNA vaccines, which deliver genetic instructions for host cells to produce pathogen proteins internally, leading to robust antibody and T-cell responses. Adjuvants are critical additions that amplify immune recognition. Aluminum salts and newer formulations activate innate sensors, creating the danger signals necessary for strong adaptive activation. Without adjuvants, subunit antigens may be recognized as harmless and fail to elicit durable responses. Boosters are repeated doses that re-expose the immune system, reinforcing memory B cells and plasma cells, and ensuring long-term antibody levels. The goal of vaccination is not only individual protection, but also community benefit. Herd immunity arises when enough people are immune, reducing circulation of pathogens and protecting those who cannot be vaccinated. This principle explains the dramatic decline of diseases like polio and smallpox in vaccinated populations, yet lapses in coverage can permit resurgence, reminding us that memory must be maintained at the community level. Vaccines also exploit immune specialization. Conjugate vaccines link poorly immunogenic polysaccharides to carrier proteins, engaging T-cell help and producing stronger memory in infants. Vector-based vaccines deliver antigen through harmless viruses, stimulating robust responses. Each design adapts to challenges posed by different pathogens. Consider a clinical vignette. An adult traveler receives vaccination against hepatitis A before visiting an endemic area. The vaccine induces specific IgG antibodies and memory T cells, preventing illness upon exposure. If instead the individual developed jaundice, fever, and abdominal pain during travel, this would require professional assessment, as it may signal infection despite prevention efforts. The vignette illustrates how vaccination shifts risk, but does not replace vigilance. The history of vaccines reveals both triumph and caution. Eradication of smallpox demonstrates what global cooperation and immunologic memory can achieve. At the same time, rare adverse reactions highlight the need for safety monitoring. The principle remains: by presenting antigens in safe form, the immune system learns without suffering disease. Reflection: vaccination is rehearsal. It allows your defenses to practice in safety so that when the curtain rises, the performance is strong and swift. In the city you have been following, rehearsals are complete, yet sometimes defense is not just prevention but therapy, intervening to dampen or redirect the immune system once disease has begun. How can modern medicine harness immunity as treatment? In the city, defense can sometimes turn into harm, persistent inflammation, autoimmunity, or unchecked cytokine release. Modern medicine has learned not only to stimulate immunity but also to guide or restrain it. This deliberate manipulation is called immunotherapy, treatment that harnesses or alters immune mechanisms to fight disease. One approach uses monoclonal antibodies to neutralize inflammatory cytokines. Anti-tumor necrosis factor, anti-TNF agents bind TNF-alpha, reducing its ability to drive chronic inflammation in conditions such as rheumatoid arthritis or inflammatory bowel disease. Anti-interleukin-6, anti-IL-6 therapies target the receptor for IL-6, dampening systemic inflammation. Anti-IgE antibodies reduce allergic reactivity by binding free IgE, preventing it from attaching to mast cells. Each therapy demonstrates how blocking a single mediator can rebalance immune activity. Checkpoint inhibitors represent another major strategy. T cells carry inhibitory receptors such as CTLA4 and PD1, which normally prevent excessive activation. Many tumors exploit these pathways by upregulating ligands that engage PD1 or CTLA4, silencing T cell responses. Antibodies that block these checkpoints release the brakes, allowing T cells to attack cancer. This approach has transformed treatment of melanoma and other cancers, yet it carries risks. Unleashed T cells may also target healthy tissues, causing autoimmune-like side effects. Cell-based immunotherapies push boundaries further. Chimeric antigen receptor T cells, KRT, are engineered from a patient's own T cells, modified to express receptors that recognize tumor antigens. When reinfused, they target malignant cells directly. KRT therapy has achieved striking remissions in certain leukemias and lymphomas, though cytokine release syndrome and neurotoxicity remain challenges. The therapy illustrates both power and peril of redirecting immunity. Vaccines, too, can be therapeutic. Cancer vaccines aim to stimulate immunity against tumor-associated antigens, though success is limited by tumor evasion strategies. Oncolytic viruses, engineered viruses that infect and kill cancer cells, also stimulate local immune responses. These therapies seek to make the immune system recognize cancer not as self but as altered tissue requiring elimination. Beyond cancer, immunotherapy addresses allergies and autoimmunity. Allergen immunotherapy introduces gradually increasing doses of allergens under controlled conditions, teaching tolerance and reducing hypersensitivity. Regulatory T-cell-based therapies are under investigation, aiming to restore balance in autoimmune disease. Each approach reflects an attempt to tune responses rather than simply suppress them. Consider a clinical vignette. A person with severe asthma, uncontrolled by standard therapy, begins treatment with an anti-IgE antibody. Over months, attacks diminish, illustrating how targeted immunotherapy can reshape disease trajectory. Yet if new systemic symptoms appear, fever, rash, or severe fatigue, professional assessment is essential, as unintended immune shifts may occur. Reflection immunotherapy is proof that the immune system is not only a source of disease, but also a tool for healing. It is the art of turning the city's own forces inward against the very problems they once created or failed to control. In your imagined city, forces have been restrained, redirected, and re-energized. But what happens when the defense must accept foreign structures, an organ or tissue not originally its own? That test of tolerance is transplantation. In the city you are following, sometimes a whole district must be replaced, a kidney, a heart, or bone marrow itself. This is transplantation, the transfer of cells, tissues, or organs from one individual to another. The challenge is that the immune system excels at distinguishing self from non-self, and transplanted material almost always bears differences. Immunity interprets these differences as threat, unless carefully managed. Central to transplantation is the human leukocyte antigen HLA system, the human version of MHC. HLA molecules present peptides to T cells, shaping recognition. Because HLA genes are highly variable, each person's combination is nearly unique. The closer the match between donor and recipient HLA, the lower the risk of rejection. Perfect matches are rare outside identical twins, but partial matches can suffice with immunosuppression. Rejection occurs in distinct timelines. Hyperacute rejection happens within minutes to hours when pre-existing antibodies in the recipient bind donor antigens, often against mismatched ABO blood group antigens. This triggers complement activation and rapid thrombosis, causing immediate graft failure. Acute rejection develops days to weeks after transplant, driven by recipient T cells recognizing donor HLA as foreign. It involves cytotoxic T cells infiltrating the graft and helper T cells releasing cytokines that recruit macrophages. Chronic rejection unfolds over months to years, characterized by progressive fibrosis and narrowing of graft vessels, eventually impairing function. The mechanism combines persistent low-grade immune attack and tissue remodeling. To prevent rejection, immunosuppressive strategies are essential. Drugs that inhibit T-cell activation by blocking calcineurin co-stimulation or cytokine signaling form the foundation. Corticosteroids reduce inflammation broadly. Antibodies against T-cells or their receptors may be used during induction phases. The balance is delicate. Too little suppression permits rejection. Too much leaves the patient vulnerable to infection and malignancy. Bone marrow or hematopoietic stem cell transplantation adds another layer of complexity. Here immune cells themselves are transferred. Instead of recipient versus donor rejection, donor immune cells may attack recipient tissues, a condition called graft versus host disease, GVHD. It can affect skin, liver, and gut, ranging from mild to life-threatening. At the same time, a degree of graft versus leukemia effect may be beneficial, with donor T cells eliminating residual malignant cells. Thus, even in conflict, there can be advantage. Consider a vignette. A patient receives a kidney transplant and initially shows improved renal function. Weeks later, creatinine rises and biopsy reveals lymphocyte infiltration, suggesting acute rejection. Professional care involves adjusting immunosuppression to rescue the graft. By contrast, a well-matched transplant with stable function demonstrates how carefully tuned immunity can coexist with foreign tissue for years. Reflection: transplantation reveals both the power and rigidity of self-recognition. To accept another's tissue requires negotiation, restraint, and constant vigilance. In your imagined city, new districts have been added, sometimes embraced, sometimes attacked. But what if the invader is not foreign tissue, but the body's own rogue cells growing uncontrollably? The next story is of immunity against tumors. In the city you have been mapping, not all threats arrive from outside. Sometimes a resident cell accumulates mutations, ignores growth controls, and expands as a clone. This is cancer. The immune system has mechanisms to detect and eliminate such rogue cells, a process called tumor immunosurveillance. Yet tumors also evolve strategies to evade these defenses, creating a constant contest between vigilance and escape. At the heart of surveillance are cytotoxic CD8 T cells and natural killer NK cells. Tumor cells often display abnormal peptides on MHC I, derived from mutated or overexpressed proteins. CD8 T cells recognize these neoantigens and induce apoptosis. If tumors downregulate MHC I to avoid recognition, they become more visible to NK cells, which are activated by missing self. NK cells release perforin and granzymes, killing abnormal cells directly. Helper CD4 T cells contribute by secreting cytokines such as interferon gamma, which activates macrophages and enhances antigen presentation. Macrophages themselves can kill tumor cells through reactive oxygen species and nitric oxide. B cells may generate antibodies against tumor antigens, which recruit complement and NK cells via antibody-dependent cellular cytotoxicity. Together, these arms form a layered defense. Tumors, however, develop escape strategies. They may secrete immunosuppressive cytokines like transforming growth factor beta or interleukin 10, which dampen effector T cell activity. They may recruit regulatory T cells and myeloid-derived suppressor cells to create an inhibitory microenvironment. Expression of checkpoint ligands, such as PD-L1, allows tumors to engage PD-1 receptors on T cells, inducing exhaustion and loss of function. Antigen loss variants, cells that no longer express immunogenic proteins, can also emerge, escaping detection altogether. This dynamic interplay is termed immunoediting, consisting of three phases: elimination, equilibrium, and escape. During elimination, immune cells destroy many tumor cells. In equilibrium, immune pressure controls but does not eradicate, selecting for resistant clones. In escape, these resistant cells grow unchecked, forming clinically detectable cancer. This evolutionary process explains both the initial absence of disease and later emergence of aggressive tumors. Modern medicine has sought to restore antitumor immunity. Checkpoint inhibitors, antibodies blocking CTLA4 or PD-1 pathways, revive exhausted T cells, leading to durable remissions in some cancers. CAR-T therapy engineers T cells to target specific tumor antigens, achieving striking results in certain leukemias. Therapeutic vaccines and oncolytic viruses aim to enhance recognition, yet with these advances come risks. Unleashed immune responses can produce autoimmunity or cytokine storms. The challenge remains to balance power with safety. Consider a vignette. A person with melanoma receives anti-PD-1 therapy. T cells reinvigorated by blockade infiltrate the tumor, shrinking lesions. Weeks later, the patient develops colitis, reflecting immune activation against normal tissues. Professional management is required to balance tumor control with tolerance. This illustrates how enhancing surveillance can uncover both benefit and collateral damage. Reflection. Antitumor immunity is a mirror, your defenses watching your own cells, distinguishing loyalty from betrayal. It is vigilance turned inward. In your imagined city, traitorous citizens have been confronted and sometimes subdued. Yet to identify them, the city needs laboratories and metrics tools to measure and analyze the hidden work of its defenders. How do clinicians assess the immune system in practice? The city cannot govern without records, registries, or archives. For an immune system that operates invisibly, the records come from laboratory studies. They are the windows through which we observe the defenders in motion, sometimes silently circulating, sometimes clashing with invaders, sometimes falling short of their task. Laboratory evaluation of immunity is not a single test, but a panorama of measurements that, taken together, describe numbers, proportions, functions, and failures. Through these, physicians recognize infection, autoimmunity, immunodeficiency, or malignancy. The first record most often consulted is the complete blood count with differential. In a small volume of blood, millions of cells are counted and classified. Total white blood cells provide an overall measure of immune presence. Within that total, neutrophils usually dominate, reflecting their role as first responders. A rise in neutrophils, neutrophilia, commonly signals acute bacterial infection or stress responses such as corticosteroid release. A fall, neutropenia, may follow chemotherapy, marrow suppression, or severe systemic infection where neutrophils are consumed faster than replaced. Lymphocytes, second in proportion, rise during viral infections, reflecting expansion of adaptive cells, and fall during immunosuppression from drugs, HIV, or malnutrition. Monocytes expand during chronic inflammation and recovery phases, eventually differentiating into macrophages. Eosinophils, though normally scarce, rise in parasitic disease or allergic states, while basophils, the rarest, occasionally increase in allergic or myeloproliferative conditions. This distribution is like a quick sketch of the immune army, showing which battalions are mobilized and which are depleted. Patterns within this differential offer deeper meaning. A left shift describes the presence of immature neutrophils, suggesting marrow release in response to infection or stress. Absolute lymphocyte counts below certain thresholds signal impaired adaptive capacity. Combined cytopenias, reductions across several lineages, suggest marrow disorders or systemic disease. Thus, a single test read carefully is not just numbers, but a dynamic story of production, mobilization, and consumption. Beyond cell counts lies the humoral arm, and here quantitative immunoglobulins are the primary measure. Antibodies are divided into IgG, IgA, IgM, IgE, and IgD, each with distinct distributions and functions. Measurement of their concentrations provides clues to deficiencies or dysregulation. Low IgG is particularly significant as IgG dominates systemic defense against encapsulated bacteria. Common variable immunodeficiency often presents with decreased IgG and poor vaccine responses, leading to recurrent sinopulmonary infections. Absence of IgA is the most common primary antibody deficiency. Most individuals remain asymptomatic, but some experience recurrent mucosal infections or autoimmune tendencies. Elevated IgM may reflect early responses, but persistently high levels can suggest hyper-IgM syndromes where class switching fails. Ig elevation indicates allergic disease or parasitic infection, sometimes extreme and rare syndromes. IgD remains enigmatic, largely serving as a receptor on naive B cells. Specialized tests examine immunoglobulin quality as well as quantity. Serum protein electrophoresis separates proteins by charge and size, producing a pattern where polyclonal immunoglobulins form a broad gamma region. A sharp spike suggests monoclonal production, as seen in plasma cell disorders like multiple myeloma. Immunofixation further defines the exact heavy and light chain composition. These tools show how humoral responses may shift from broad polyclonality to malignant monoclonality. Serology bridges humoral presence with specific targets. By detecting antibodies against defined antigens, serology answers questions of exposure, infection, and autoimmunity. For infections, IgM indicates recent or current exposure, while IgG signals past infection or immunization. In hepatitis, detection of surface antibodies marks immunity, while core IgM suggests acute infection. In syphilis, treponemal and non-treponemal tests are combined to distinguish active disease from past exposure. For autoimmunity, anti-nuclear antibodies ANA provide a broad screen, positive in many connective tissue diseases. Subtypes sharpen focus. Anti-dsDNA and anti-Smith are highly specific for lupus. Anti-CCP points to rheumatoid arthritis. Anti-SSA and anti-SSB indicates Sjogren's syndrome. Interpretation requires caution, as low-level positives occur in healthy individuals, and context determines significance. Serology is thus not a definitive verdict, but an interpretive language requiring nuance. Complement assays extend the analysis. The complement system, a cascade of plasma proteins, is central to innate immunity and cooperative with antibodies. Measuring its activity provides insight into both genetic deficiencies and acquired consumption. The CH50 assay tests the classical pathway by measuring hemolysis of antibody-coated red cells. Absence indicates failure in any early component. The AH50 assay tests the alternative pathway. Low levels suggest active immune complex disease as complement proteins are consumed during inflammation. In lupus nephritis, for example, low C3 and C4 levels mark ongoing deposition and activity. Isolated deficiencies in late components predispose to recurrent infections with Neisseria. Thus, complement testing both diagnoses inherent absence and tracks disease activity in immune complex disorders. Flow cytometry offers a detailed census of immune cells using fluorescent antibodies that bind surface markers. This technique identifies T cells, B cells, NK cells, and their subsets. It can distinguish naive from memory populations, helper from cytotoxic subsets, and regulatory T cells from effectors. In immunodeficiency, flow cytometry may reveal absent B cells in X-linked gammaglobulinemia or reduced CD4 counts in HIV. In hematologic malignancies, it defines abnormal clonal populations, distinguishing leukemia or lymphoma types. Intracellular staining adds further depth, showing cytokine production or transcription factor expression. Flow cytometry is not just counting but mapping phenotypes and functions in exquisite detail. Functional assays probe performance directly. The neutrophil oxidative burst test assesses the capacity of phagocytes to generate reactive oxygen species, essential for killing engulfed microbes. Defects identify chronic granulomatous disease, where catalase-positive organisms proliferate unchecked. Lymphocyte proliferation assays expose cells to mitogens or specific antigens and measure division, revealing T-cell competence. NK cell cytotoxicity assays measure the ability to lyse susceptible targets. Together, these assays move beyond presence to capability, showing whether defenders can execute their roles effectively. Molecular methods expand possibilities. Polymerase chain reaction detects pathogen DNA or RNA at extraordinary sensitivity, identifying infections before cultures grow. This has revolutionized viral diagnosis, from HIV load monitoring to influenza and SARS-CoV-2 detection. Sequencing immunoglobulin or T-cell receptor genes reveals clonality, tracking malignancies or residual disease after therapy. Next-generation sequencing can profile entire immune repertoires, mapping diversity and skewing. Cytokine profiling using multiplex bead arrays or microfluidics quantifies dozens of mediators simultaneously, painting portraits of inflammatory states in autoimmune disease or cytokine storm syndromes. These tools approach the frontier between clinical care and research, yet increasingly inform decisions in immunotherapy. Clinical interpretation is always integrative. Numbers alone mislead unless patterns are considered. A patient with recurrent bacterial infections, low IgG, and absent vaccine responses likely has antibody deficiency. A patient with fever and profound neutropenia after chemotherapy is at immediate risk of sepsis despite normal antibody levels. A patient with high ANA titers, anti-dsDNA positivity, and low complement demonstrates active lupus, while similar ANA without symptoms may be benign. Thus, laboratory tests are not verdicts but pieces of puzzles, assembled by context. Case vignettes illustrate a young woman with fatigue, arthralgia, and rash is found to have anemia, positive ANA, and low complement. The pattern suggests lupus, highlighting autoantibody production and immune complex consumption. Another patient with recurrent sinus infections has absent IgA, but otherwise normal immunity, consistent with selective IgA deficiency. Each scenario underscores how laboratory studies translate immune dynamics into clinical narratives. Laboratories also monitor therapies. In patients on immunosuppressants, lymphocyte counts track suppression. In HIV, CD4 counts and viral loads guide management. In biologic therapies targeting cytokines, monitoring inflammatory markers ensures safety and efficacy. Immunotherapy trials employ cytokine profiling and flow cytometry to detect activation or exhaustion states. Thus, tests not only diagnose but also guide treatment and safety. In the metaphor of the city, laboratories are observatories. From towers overlooking the walls, analysts count the soldiers, record their weapons, and track their movements. They detect shortages, surges, or abnormalities that foretell trouble. They cannot themselves defend, but they provide the knowledge without which defense would be blind. Reflection: laboratory studies are translations, turning invisible molecular battles into numbers, graphs, and images. They do not capture every detail, but reveal enough to interpret patterns and intervene. They are the language of the unseen, and through them, the hidden city becomes legible. In the city you have been following, the laboratories now report on strengths, weaknesses, and silent battles. Yet numbers alone cannot convey the urgency of lived cases. How do these principles appear when the patient in front of you is short of breath, febrile, or collapsing? That is where principles become stories, and where the next chapter begins. The city's map has grown vast. Barriers, patrols, couriers, signals, and laboratories all described. Yet maps alone cannot convey the urgency of decisions at the bedside when someone suddenly gasps for air, collapses with fever, or develops pain without clear cause. Clinical pearls are condensed lessons drawn from lived cases, each showing how immune principles translate into urgent realities. They are reminders that the immune system is not abstract, it is embodied in lives where choices matter. One striking scenario is anaphylaxis, a systemic type 1 hypersensitivity reaction. Imagine a person with peanut allergy taking a single bite of a cookie. Within minutes, mast cells primed with IgE antibodies release histamine, leukotrienes, and prostaglandins throughout the body. Blood vessels dilate, plasma leaks into tissues, and airway smooth muscle contracts. The sequence is rapid, first itching, then hives, then swelling of lips and eyelids. Breathing becomes difficult, wheezing emerges, and blood pressure falls. This is not gradual immunity, but an overwhelming wave. The pathophysiology is IgE cross-linking on mast cell surfaces, a mechanism designed to repel helminths but misapplied to harmless proteins. The pearl is that a sudden severe reaction to food, insect sting, or drug must be recognized as an emergency. Delay can mean collapse. Recognition of patterns, rapid onset, multi-system involvement is crucial. A different but equally dangerous lesson comes from febrile neutropenia. A person receiving chemotherapy for leukemia develops profound bone marrow suppression. Neutrophils, normally numbering in thousands, drop to single digits. A fever arises, sometimes the only sign. Without neutrophils, bacterial containment fails. Infections that would have remained local spread through the bloodstream, seeding lungs, liver, and skin. Even minor complaints, mouth ulcers, mild cough, or chills may herald systemic sepsis. Here the pearl is that fever in a neutropenic patient is always urgent. The absence of innate cells creates vulnerability as dangerous as their overactivation. This case illustrates that defense requires presence, not only restraint. Autoimmunity provides its own lessons. Consider a young woman with joint pain, low-grade fever, and a malar rash across her cheeks. Laboratory tests reveal anti-dsDNA antibodies and low complement, reflecting immune complex deposition. The mechanism is type 3 hypersensitivity. antibody-antigen complexes depositing in tissues, activating complement, recruiting neutrophils, and damaging organs. The pearl is that systemic autoimmune disease presents with protean symptoms but leaves fingerprints, autoantibodies, and complement consumption that distinguish flare from infection. This distinction is vital. Treating a flare with immunosuppression versus treating an infection with antimicrobials can mean opposite paths of care. Other pearls come from absence of immune organs. A splenectomized patient years after trauma presents with overwhelming sepsis from Streptococcus pneumonia. The missing spleen explains the absence of rapid antibody responses against polysaccharide capsules and the loss of splenic macrophages that normally filter bacteria from blood. The lesson is that a single organ, often taken for granted, plays an irreplaceable role in defense. Immunodeficiency in children offers profound insights. A toddler with recurrent thrush, pneumonia, and chronic diarrhea is found to have severe combined immunodeficiency. Both T and B cells are absent, leaving no adaptive defense. Infections pile up relentlessly. The pearls that absence of adaptive immunity reveals how much of daily survival depends on silent memory and specific recognition. These rare but severe cases are windows into how essential normal immunity is for life. Other pearls illustrate the costs of imbalance. A patient with uncontrolled asthma shows how THET2-driven IgE responses, useful against parasites, can misfire against dust mites. A farmer develops hypersensitivity pneumonitis from inhaling moldy hay, a type 3 immune complex disease manifesting as lung inflammation. A traveler with malaria demonstrates how intense cytokine release, particularly TNF-alpha, produces fever, rigors, and sometimes cerebral complications. Each scenario shows immune principles in action, each reinforcing that mechanisms studied in textbooks are visible in clinic rooms. Pattern recognition is the unifying thread. Anaphylaxis teaches that mast cells can unleash systemic mediators in minutes. Febrile neutropenia shows that absence of defenders is as dangerous as their excess. Lupus illustrates the double-edged nature of antibodies, protective when directed outward, damaging when directed inward. Splenectomy demonstrates the silent contribution of a single organ. Severe combined immunodeficiency exposes the bedrock role of adaptive memory. Together, these pearls become a mental library, guiding clinicians to interpret signals quickly. More pearls enrich the library. Serum sickness-like reactions remind us that immune complexes can be induced by drugs as well as infections. Hemolytic disease of the newborn shows maternal IgG crossing placenta, destroying fetal red cells. Chronic granulomatous disease, where neutrophils cannot mount oxidative burst, teaches that defects in microbicidal machinery allow unusual pathogens to thrive. Each pearl distills a principle into a memorable clinical image. Consider a case vignette. A middle-aged man develops fever and painful swollen knee after a minor scrape. Synovial fluid grows Staphylococcus aureus. The lesson is that joint infections, though sometimes subtle, can rapidly destroy cartilage and bone. The pearl is that local inflammation plus systemic fever is a warning sign not to ignore. Another vignette. A child presents with eczema, recurrent staphylococcal infections, and high IgE levels. This triad points to hyper-IgE syndrome, illustrating how overactive pathways create paradoxical vulnerability. Reflection. Real lives make mechanisms vivid. Behind every term IgG, complement, neutrophil, are faces, voices, and moments where immune balance decided the outcome. These pearls are not simply facts, but bridges between invisible molecules and visible suffering or relief. In your imagined city, you have now seen defenses misfire, collapse, and recover. You have walked through sudden storms, silent absences, and persistent misdirections. The tour nears its end. What remains is a quiet summary, a chance to trace the arc from innate barriers to adaptive memory and to close with calm. The city is quiet again, gates are closed, watchtowers dim, and patrols return to their stations. Calm descends after long cycles of alarm and defense. To understand this stillness, it is worth tracing once more the whole journey of the immune system, from the first blunt barriers that meet every traveler to the refined memory that remains long after each battle. The beginning is deceptively simple. At the outer boundary lies skin, a keratinized shield whose dryness, acidity, and constant turnover deny footholds to most microbes. Its sweat and sebum add chemical barriers. Mucosal layers provide softer but equally vigilant defenses. The respiratory tract lines itself with cilia, brushing particles upward and out, while mucus traps intruders. The gastrointestinal tract surrounds food with acidity in the stomach and enzymes in the intestine, while secretory IgA coats the lumen, intercepting pathogens before they reach epithelial cells. Commensal microbes, diverse and numerous, form a living curtain, competing for nutrients and space, producing antimicrobial molecules and teaching immune cells the difference between friend and foe. These barriers are not static walls, they are living fortifications, constantly renewed, constantly adapting. Breaches here through wounds, burns, or antibiotic disruption of microbiota open pathways for deeper invasion. Once a breach occurs, innate defenders mobilize. Neutrophils, short-lived but numerous, are the first to swarm, guided by gradients of chemokines and activated complement fragments. They engulf microbes, release proteases, and form extracellular traps of DNA studded with enzymes. Macrophages, resident in tissues, recognize invaders through pattern recognition receptors. They phagocytocyte microbes, release pro-inflammatory cytokines, and present antigens. Natural killer cells patrol for missing self, detecting cells that downregulate MHC I in attempts to escape cytotoxic T cells. They release perforin and granzymes, inducing apoptosis without prior sensitization. This rapid response is coordinated by receptors tuned to danger. Pattern recognition receptors, such as toll-like receptors, sense conserved microbial motifs, lipopolysaccharide, flagellin, unmethylated DNA, and damage-associated signals from injured cells. Their activation leads to cascades of transcription factors, the release of cytokines, and the orchestration of inflammation. Vessels dilate, permeability increases, leukocytes adhere and migrate. The classical signs of inflammation, redness, warmth, swelling, and pain, are ancient reflections of vascular and cellular events. Complement amplifies these early defenses. Activation by antibodies, microbial surfaces, or lectins leads to cleavage of C3 and C5, generating opsonins, anaphylatoxins, and membrane attack complexes. A few molecules of recognition expand into waves of lysis and recruitment. This principle of amplification, where small beginnings yield large effects, characterizes the entire immune system, yet amplification alone cannot sustain balance. Without precision or memory, innate defenses risk exhaustion and collateral damage. The transition to adaptive immunity comes through antigen presentation. Dendritic cells engulf microbes and migrate to lymph nodes, displaying peptides on MHC molecules. In the paracortex of nodes, naive T cells scan these displays. If the right receptor engages and co-stimulatory signals confirm danger, the T cell activates. CD4 helper T cells differentiate into subsets shaped by cytokines. The E1 cells produce interferon gamma to activate macrophages. The E2 cells release IL-4 and IL-5 to support B cells and eosinophils. The E117 cells drive neutrophil recruitment through IL-17, and regulatory T cells dampen responses. CD8 cytotoxic T cells expand, armed to kill infected cells precisely. B cells, stimulated by antigen and helper T cells, proliferate and differentiate. In germinal centers, they undergo class switching and affinity maturation, refining their antibodies. Early IgM responses give way to IgG for systemic defense, IgA for mucosal protection, IgE for parasites and allergies. Plasma cells, short-lived in lymphoid tissues or long-lived in marrow, secrete antibodies that circulate widely, neutralizing toxins, opsonizing bacteria, and activating complement. Lymphoid organs provide the architecture for these interactions. Bone marrow generates precursors, thymus trains T cells through selection, lymph nodes collect antigens from tissues, spleen filters blood, mucosal tissues monitor airways and intestines. Traffic between these sites is continuous, guided by chemokines and adhesion molecules. Without this structured movement, rare antigen-specific cells would never encounter their cognate targets. The organs are both classrooms and marketplaces, where instruction, selection, and exchange take place. Cytokines form the language of coordination. IL-1, IL-6, and TNF-alpha raise fevers, induce acute phase proteins, and recruit neutrophils. Interferons set antiviral states. IL-4, IL-12, and IL-23 polarize T cells. IL-10 and TGF-beta restore calm. When balance fails, storms erupt, overwhelming tissues and leading to shock. Thus, cytokines demonstrate both necessity and danger, the need to speak clearly and the harm of shouting too loudly. Tolerance protects the self. Central tolerance deletes or edits strongly self-reactive clones in marrow and thymus. Peripheral tolerance relies on regulatory T cells, anergy, and checkpoints like CTLA4 and PD-1. Failures produce autoimmunity, lupus with immune complex deposition, rheumatoid arthritis with joint destruction, Graves' disease with thyroid stimulation. Hypersensitivities show other misfires: IgE against pollen, IgG against red cells, immune complexes against serum proteins, T cells against contact antigens. Deficiencies reveal fragility: absence of B cells in agamoglobulinemia, absence of T cells in SCID, depletion of CD4 cells in HIV, loss of spleen after trauma. Vaccination represents deliberate training: live attenuated, inactivated, subunit, conjugate, and mRNA vaccines all provide safe exposure, teaching memory without disease. Adjuvants amplify signals and boosters reinforce them. Immunotherapies refine the system further, dampening inflammation with anti-TNF or anti-IL-6, blocking IgE to control allergy, or unleashing T cells against tumors with checkpoint inhibitors. KRT therapies re-engineer cells for precision attack, while allergen immunotherapy retrains tolerance. Transplantation challenges self-recognition most directly. Donor organs display different HLAs, triggering hyperacute, acute, or chronic rejection. Immunosuppressive regimens attempt to balance graft acceptance with infection risk. In bone marrow transplants, donor cells may attack the host in graft versus host disease, while at the same time providing graft versus leukemia benefit. Tumor immunity demonstrates surveillance turned inward. Cytotoxic T cells and NK cells detect transformed cells, yet tumors evade by downregulating antigens, secreting inhibitory cytokines, or expressing checkpoint ligands. Immunoediting describes elimination, equilibrium, and escape. Therapies seek to restore balance, but risk autoimmunity in the process. Laboratory tools reveal these dynamics: blood counts, immunoglobulins, serology, complement activity, flow cytometry, functional assays, and molecular techniques all translate invisible battles into measurable data. Clinical pearls embody them: anaphylaxis showing mast cell overreaction, febrile neutropenia showing vulnerability from absence, lupus flare showing immune complexes misdirected, splenectomy showing the cost of missing organs, immunodeficiency showing the need for adaptive responses. Across all these layers, one theme emerges: the immune system is not a single weapon, but a symphony of defenses, sensors, and signals. It is fast when neutrophils swarm, slow when antibodies mature, broad when cytokines flood, precise when T cells target, destructive when storms rage, reparative when tolerance restores calm. It must protect without destroying, remember without overreacting, and adapt without losing balance. To know this system is to value restraint as much as force, memory as much as immediacy. The quiet you imagine now is not absence but readiness, a poised equilibrium. Defenses remain awake even in stillness. Cells continue to circulate, antibodies continue to drift, signals continue to whisper. The system works unseen, carrying out its vigilance while you rest. Reflection: the immune system is the unseen companion of life, balancing alertness with peace, strength with gentleness. Its vigilance allows calm, its memory allows trust. In your imagined city, the lights dim. Patrols remain but move quietly, couriers whisper instead of shouting, laboratories close their books. The journey has shown how innate barriers hand off to adaptive memory, how signals orchestrate, and how balance preserves health. What remains is calm awareness, a system always awake, rarely noticed, yet endlessly faithful in its watch. The city of immunity now lies quiet under a twilight sky. Streets that once filled with alarms, rushing patrols, and secret couriers have settled into calm order. The gates are closed but not locked. Sentinels remain on watch, their eyes steady but untroubled. You have walked through the layers of defense, from the outer barriers that greet every visitor to the inner chambers where memories are written. You have seen how cells of different lineages converge, each with a role distinct yet connected, some rushing to inflame, others stepping in to repair, still others waiting in silence until needed again. The system is vast, but its rhythm is steady, repeating every day without your awareness. In quiet reflection, notice that most of the time this orchestra plays unheard. Neutrophils circulate, antibodies drift through fluids, lymph nodes filter, and cytokines speak in whispers. You feel none of it because most of it works without error. The times you do notice, fever, swelling, fatigue, are only the crescendos, the visible signals of hidden work. There is reassurance in this. Even while you rest, immune patrols adjust, balance, and reset. Cells that fought today are replaced. Those that learned will remember. Your health depends less on constant vigilance from you, and more on their unbroken rhythm, refined over lifetimes and generations. So as the lights dim further in your imagined city, allow its quiet steadiness to become your own. Defenses are active but calm, prepared but silent. You can let go of the maps and the battles because they will continue without you needing to intervene. This journey ends here, not in alarm but in peace. The immune system continues its watch, unseen and unceasing, while you step back from the city walls and find rest.